In this tutorial, we're going to look at plant growth hormones. The first aim is, can you describe the application of plant growth hormones? Then can you explain how auxins cause growth in shoots and the roots of plants? And finally, can you interpret experimental data on plant growth hormones? Now, I know often the blue whale is given credit for being the largest living thing on this planet, but actually plants would really take the crown. If you ever have the fortune of visiting the redwood forests in California, you'll see huge sequoia trees which are literally giants of the natural world. So today's picture is of a wisteria. Wisterias are beautiful flowering plants. There is a Japanese wisteria which is 144 years old, and it's actually grown to the size of half an acre. That's almost 2,000 meters square, phenomenally large and incredibly beautiful. So plants are quite commonly labelled by students as boring, but they are anything but, and in another tutorial on photosynthesis you'll see why. So plant growth hormones are basically chemical messages that bring about growth in plants. There are two types of hormones you need to know about. Firstly, auxins, which stimulate growth in roots and shoots of plants. And then gibberellins, which stimulate stem growth, seed germination and flowering in plants. So we can use these plant growth hormones to maximize profit when cultivating plants. So firstly, we can use plant growth hormones as selective weed killers. So weeds generally have broader leaves than, say, cereal crops. So when we spray a field of crops, including the weeds, with a specific hormone, the hormone targets the broadleaf plants and basically destroys the weeds without damaging the cereal crops. Next, we can quite easily clone plants with desirable features using their cuttings and rooting powder, which contains a plant growth hormone. So what you do first is you would cut an appropriate part of the plant. Let's see where it branches off from this central stem. You then take the cutting and dip it in some rooting powder, and then you plant the cutting back into some soil, and the rooting powder will stimulate growth in the roots, and you'll get a clone of the original plant. You can also use plant growth hormones to control the ripening process. So you could either delay the ripening of plants or fruits, for example, to increase their shelf life, but also you could pick the most unripe fruits. And remember, when plants are unripe, they are tougher and they will basically be more resistant to damage during transport. You can then spray them with the appropriate hormone and stimulate ripening, so by the time they get to the supermarkets, they're ripe. Obviously, when a fruit is picked in a ripe state, then it will ripen very quickly and over-ripen before it has time to get to the supermarkets. You can also use plant growth hormones to produce seedless fruit. So think about this example of watermelon. So whenever you eat watermelon, you have to spend half your time spitting out seeds, but if you could apply hormones, then you could produce a seedless watermelon. So that is how you can describe the various applications or commercial applications of plant growth hormones. So there are just 4 to remember there. So now let's look at how auxins actually work. So firstly, we'll look at how auxins bring about growth in the shoots of plants. Step 1, auxins are produced in the shoot tips. So you can see the pink crosses there are auxins in the tip of the shoot. Next, auxins will diffuse down the shaded side of the root. So you can see sunlight coming this way, it could be any sort of light, and you can see the auxins start to diffuse down the shaded side, but not the side which is exposed to sunlight. Here they cause cell elongation. So unlike us, we grow through cell division, but plants will grow through cell elongation. So if you look at this side, you can see the auxins have diffused down this side, and where they're binding to cells, they're causing them to stretch or elongate. But the cells which aren't receiving as many auxins, they'll still receive a few, but where they're not receiving them, growth or elongation is occurring slower. So this results in a positive phototropism. Photo means light, tropism means movement or growth or bending towards, and you can see because elongation has occurred on this side, the plant grows towards the light or bends towards the light. So just to review that process, auxins are produced in the shoot tip, auxins diffuse down the shaded side of the shoot where they cause cell elongation, and this results in a positive phototropism. Be aware that a positive phototropism is a growth towards the light, whereas a negative phototropism is a growth away from the light, but plants will experience a positive phototropism in their shoots. So one of the reasons I think students find plants boring is because they seem to do everything quite slowly. But if you can use something like a time-lapse camera to speed up the process, you can see them doing things which normal living things do. So in this video, I've basically left a time-lapse camera running for a few hours to look at some chili plants grow. The next plant was actually filmed during a 100-minute science lesson, so you could actually see this movement in a space of just 100 minutes. Now let's look at how auxins bring about growth in the roots of plants. Now this only works because roots develop laterally, horizontally, not vertically like shoots. So firstly, once again, auxins are produced in the tip of this time the root. So you can see auxins are produced in the tips. Then auxins will accumulate or build up on the underside of the root. This is because of gravity acting on the auxins. They start to build up on the lower side of the root. Now this is where it gets a little bit different. Because auxins basically build up on the lower side and they keep on building up, they basically reach very, very high concentrations. And in very, very high concentrations, they do something quite different. They in fact inhibit cell elongation. So what we're getting instead is now the cells which are less exposed to auxins are elongating as normal, but the ones which are exposed to high concentrations of auxins have actually experienced less growth. So the result is a positive geotropism. Geo means ground or earth, or you could also say gravitropism, a movement towards gravity. So you can see the root starts to curve downwards where it can reach more moisture because obviously plants require water to survive. And just like before, a positive geotropism is a growth towards the earth, whereas a negative geotropism is a growth away from the earth. So plant roots experience positive geotropisms. So just a quick recap again, in roots, auxins are produced in the root tip, auxins accumulate or build up on the underside of the root at the bottom. In high concentrations, they inhibit cell elongation, so cell elongation is actually slowed down on the underside of the root, and that leads to a positive geotropism. And that is how you explain how auxins cause growth in shoots and roots of plants. So now let's look at how we investigate plant growth hormones. So in this simple experiment, we have 3 shoots growing. In one shoot, we've done nothing to it. In the second shoot, we've covered just the tip so it can receive no sunlight. And in the third shoot, we've covered a part of the stem below the tip so the tip can receive sunlight. So let's say we come back a week later to observe the results. We see shoot 1 has basically developed as we expect. We get a positive phototropism, a growth towards the light. With shoot 2, we experience no bending towards the light, but with shoot 3, we do again see that bending towards the light, that phototropism. So this experiment proves that auxins are produced in the tips of the shoots where they will respond to sunlight. In the second experiment, we get a shoot and we cut the tip from the main stem. We then get some agar jelly and we place the tip on top of the agar block so the auxins diffuse into the agar block. So now we have an agar block with a high concentration of auxins. Then we balance the agar block on one side of the shoot and we observe maybe a few days later that the shoot is now bending this way. So what this experiment allows us to do is build up a high concentration of auxins on one side of the shoot, and therefore proving that shoot growth is fastest where auxin concentration is greatest. You can also quite simply perform your own experiment to test the effect of auxins. For this, you will need 3 Petri dishes, and you put damp cotton wool at the bottom of each. Then you place 10 cress seeds in each one, and then you expose them to light sources from different directions, so from the left, from above, and from the right. If you come back 1 week later, you'll find that the cress seeds which were exposed to light source from the left will have grown towards the left. Those which were exposed to light source from above will have grown straight upwards, and those which were exposed to light sources from the right will have grown to the right. This clearly proves that plants grow towards the light. And this is obviously important to plants because it means that their leaves are now facing the light square on so they can absorb as much light as possible to drive photosynthesis, which makes food for them so they can survive and grow. A few controls to consider in this experiment are the number of seeds, so you need the same number of seeds in each experiment. The type of seeds, so obviously different seeds will have different growth rates, so make sure they're all cress, for example. The temperature of each Petri dish must be the same, and you must also account for the fact that light is a heat source, so you could use a thermometer to check that they're all the same temperature. The amount of water you produce in each one, you don't want that to limit growth, so the amount of water must be the same, and obviously the light intensity, they must be exposed to the same, if you like, concentration of light, but light intensity is a proper word. And that is how you can interpret experimental data on plant growth hormones.